Well, good afternoon, everyone. Welcome to our CCOVI lecture today on February 12th of 2013. It's my pleasure to introduce my colleague and good friend, Dr. George van der Merwe. George is Associate Professor of Molecular and Cellular Biology at the University of Guelph, but he's also a CCOVI Fellow. And we do a lot of collaborative research with George based on his level of expertise that he brings to the wine industry. George holds a Bachelor of Science in Microbiology as well as a PhD from the University of Stellenbosch in South Africa. His lab focuses on the molecular mechanisms that enable the adaptation of yeast to fermentation-related stresses, and he also links that to their molecular adaptation and metabolite production. So this becomes really important for us when we're looking to develop and select yeasts that are really primed for wine production, especially under difficult fermentation conditions, things like ice wine production or sparkling wine production, which is a new area that George is quite active in. So the knowledge generated from his research enables researchers to devise strategies to improve stress tolerance, vitality, and efficiency of yeast during performance of alcoholic fermentations. So today he's going to share with us some of his more recent research in this area.
Thanks, Debbie. It's always nice to come back to Brock. It's where it all started many years ago. I came here and I started here in 1998, so it always feels like the circle is completed when I come back here. I'm going to talk a little bit today about getting started in fermentation and the adaptation of wine yeast to early fermentation conditions. And it's actually a project that we kind of backed into rather fortuitously. It wasn't really a focus, but it came about. Just as a little bit of background, I haven't done this before, and I thought people should actually know. The research that we do in our lab, we do fundamental research and with a focus of looking at wine production. And then we focus on looking at wine yeast development, as Debbie mentioned, and with a specific focus on the molecular adaptation of the yeast to fermentation-related stresses. And then a second leg that we're busy developing now with CCOVI is to look at microbial wine spoilage, and more specifically on the yeast side of that. And our current focus that we're looking at is Brettanomyces, but I'm not going to be talking about that today at all. So what we're looking at is basically the molecular response of the yeast during fermentation. So this relates to general wine production, where you have table wine production, and that production process can be rather stressful for yeast, specifically in Canada, with the cold growth seasons and the acidic musts that we have to deal with. So that is a significant problem. Ice wine fermentations are also a significant problem, as they mentioned, and we're also getting started with sparkling wine production, specifically the secondary production, secondary fermentation, where we're actually looking at bottle fermentation, but we'll speak about that next week, I think. Yeah. So the focus today would be just to look at regular table wine production. So we all know these beasts. Many of the winemakers actually buy Saccharomyces cerevisiae or Bayanis. It buys them as actively dried yeast from a variety of different companies, and this is what they look like to you guys, to winemakers when they actually get them, and they start to get dehydrated, and they are consequently inoculated into fermentations, into grape must for wine production. Some people also do spontaneous fermentations, but by and large, many winemakers actually use this. If we actually go onto a molecular, onto the microscopic level, this is a 3D scanning electron micrograph of the budding yeast, where you can see the mother cell and the daughter cell budding off that. And then if we get into more detail with higher resolution, we can actually see the plasma membrane and the cell wall of this organism. So this microorganism was originally thought to be an enzyme that produces wine, which we thankfully now know is not an enzyme, but it's now a microorganism. And consequently, it's a live entity that actually responds to environmental conditions and to the fermentation. So what is it that the yeast actually do? So if you have a mother cell here with a daughter cell that's budding off the mother cell, so this is a growing Saccharomyces yeast, what I've actually shown here are these nice round circles that in my mind represents nutrient transporters or transporter proteins that will actually carry components from the grape must into the yeast itself. And so you get things like sugars or precursors for metabolite production that are carried physically into the cells by other proteins, by these membrane transporters. It doesn't just happen to float into the cell. This is an active process. What then happens is that this serves as a substrate typically for enzyme reactions, and then you get the production of some kind of product that would ultimately lead to wine production. So we get ethanol production and then also a variety of different flavor compounds. But the yeast also grows, so it produces biomass, other proteins, lipids, etc., macromolecules that would allow this daughter cell to grow and become bigger and consequently form a second cell. So this is by and large what the yeast is used for, but this is all underscored by a large process that is regulated or that can adapt to the different conditions in the environment. So all the genes that we have, we find in Saccharomyces cerevisiae, or in any organism, would be contained in its DNA. This gets expressed or transcribed into RNA, which would encode proteins. These enzymes that perform the conversion of the substrate into the product, or even these membrane proteins or membrane transporters that carry the nutrients and the precursors into the cells, are all proteins. So they all basically are underscored or supported by this process. Even the proteins that are being produced here for the cell to grow, the biomass, they are also proteins. So we've got this process that is basically underscoring, we call it the central dogma, that basically underscores the activity of the yeast. When the yeast goes and actually it's subjected to all the conditions that we'll speak about in a minute during wine production, then this is adapted. The level of gene expression adapts. The translation of that into proteins adapts, and also the activity of the proteins can be adjusted or adapted, or even where they're localized to in the cell can be regulated. So this is basically where the majority of the regulation takes place, would be, or the adaptation takes place, would be in this realm over here at a molecular level. So I'm going to gear down a little bit, and let's look at the optimal growth conditions for Saccharomyces cerevisiae, just to get an idea of what are we really dealing with here, right? Because it's very easy for us to go and actually dump yeast into a fermentation and off it goes, right? And so if we think about what these organisms really like, they like to grow at a temperature between 25 and 30 degrees Celsius. You hardly ever see that in wine fermentations, right? At least in the start, you hardly ever see that. The pH is typically between 5, optimal pH is between 5 and 5.5. We never see that in the Ontario grape industry, actually anywhere. It doesn't like growing in concentrations of ethanol higher than 1.4%. It's one of the few microorganisms that can tolerate high concentrations of ethanol, but it doesn't like to actually grow, in other words, form a second cell in the presence of high concentrations of ethanol. It has a preference for specific nitrogen sources like ammonia and glutamine, but it can use other amino acids as secondary nitrogen sources that are poorer in quality, and this affects its ability to actually grow effectively. It prefers to grow in the presence of oxygen, even though it is known as a facultative anaerobe. So it can grow in the absence of ethanol, but it likes to grow in the presence of oxygen. It likes to grow in the presence of oxygen because it's easier for it to make membranes in the presence of oxygen. So to construct new membrane, it needs oxygen to do that. And then it likes to grow in an environment where there aren't a lot of solutes in the environment. In other words, with a low osmotic stress, not hyperosmotic stress that we would typically find in the wine industry. So if we compare that, what the yeast likes, to what it actually experiences in reality when we actually do wine fermentations, you can see that the temperature varies, but it's typically low. Most of the white wine productions, the fermentations actually run in the teens degrees Celsius to get the fermentation to be efficient and effective. So we're typically looking at anything from 12 to 18 degrees Celsius. Red wines can be a little bit warmer, but not really at these high temperatures. If we look at pH, the pH is typically lower than 3.4, in most instances closer to 3, or between 2.8 and 3, where we're looking at pH values, especially in a colder growth season. So this is an enormous acid stress that the yeast has to cope with, in that there's a high concentration of high acidity around. When we look at ethanol that's being produced during wine fermentations, typically between 11 and 16%, depending on where you are in the world. So this is almost an order of magnitude higher that the yeast is actually finding itself in. Right? So this is not a friendly environment. Nitrogen is often present in very low concentrations, but we know winemakers supplement with nitrogen, so that isn't really a massive issue. Fermentations are anaerobic in nature, so the yeast consequently do not have access to all the oxygen to produce new membrane. And when we look at the solutes that we have within just regular grape must, we see that the water activity drops, and if you actually go, in this instance, and if you actually go into ice wine, it can actually drop further because of the higher concentration of sugar. So what we're dealing with here is an organism that's really required to do its job outside of the conditions that are supposed to be optimum for itself, for it to live in. So what do we do? What do winemakers do is we buy these active dry yeast that's typically grown in a respiratory environment. Consequently, there's a lot of oxygen around, a lot of mitochondria around, and you get a lot of membrane that's being produced. Then they're very rapidly dried, and you see them in that grainy composition that in which they are packaged. And on the side of this very nice package, you see something that's typically like this. Or from another distributor, you see a written out little recipe of what you're supposed to be doing. So you add this dried yeast to warm water or some warm fluid, typically water between 35 and 40 degrees Celsius, to rehydrate mainly the membranes of those yeast so that they can become active again. Then they ask you to actually sit this down, stir it and let it sit for about 20 minutes, and then consequently wait for it to actually rehydrate. And then you've got to wait until the temperature is within 10 degrees of your juice that you're inoculating it into. There's a reason for that, is that the yeast has to actually adapt to the new temperature it's going to go into. If you're at the 10 degrees difference at the top end of that, it is still in the process of adapting, and it will go through a significant lag phase because it has to adapt to a new temperature that it's going to be going into. If you shift the cells from 38 degrees Celsius into something that's 12 or 18 degrees Celsius, there's a massive adaptation that the yeast has to go through. As it is, it will go through that process anyway, but it's going to take them a little bit shorter if you actually wait for the juice to cool down, or for the inoculant to cool down. This basically just takes care of a little bit of the temperature adaptation that it has to go through. If we look at the typical wine fermentation, right at the start of the fermentation, we've got a low pH, as we said before. Typically, the juice is in low temperature. We're typically looking at something at about 15 degrees Celsius. Oxygen is typically not much around because most winemakers protect their juice. They don't want oxygen around to protect against oxygenation of the flavor compounds, so they try to limit that to the lowest possible level. And you've got this hyperosmotic stress. So these are all the things that that active dry yeast has to deal with once it's actually coming out of its rehydration process immediately into this stage. So what we then have here is that we've got on the y-axis you've got say 22 degree Brix concentration sugar grape must here. And then with these characteristics, that's part and parcel what grape must would look like. The yeast would actually grow. This would be the biomass that's being produced. So this is the amount of growth that the yeast would go through while it starts to use this sugar. So this sugar is basically converted into biomass, in other words, into the production of new Saccharomyces. It's growing at that stage. It's not really producing a lot of ethanol yet. It's growing. And in most instances, the growth phase is very short in a wine fermentation. It'll go through maybe 5 or 6 growth cycles and then it'll stop. And the reason for that is basically the ethanol that's being produced. It's starting to produce, and that would inhibit cell cycle progression. In other words, inhibit growth to a large extent. So you see this rapid growth up to a certain point. There's still a massive amount of sugar left here. But basically the yeast is entering into stationary phase, meaning that it's not producing another daughter cell, it's not growing actively anymore. Notice also that the ethanol concentration is fairly low at this point. While it is actually in stationary phase, it starts to consume the sugars. It's in stationary phase, it's not growing actively, but it's metabolically active. In other words, all the enzymes that we spoke of earlier are still active, and the yeast can therefore perform its function and convert these sugars into ethanol. It's not growing that much anymore, but the yeast is still viable, and consequently it consumes the sugar, produces ethanol, and the ethanol concentration actually increases. We'll talk about ethanol tolerance a little bit later. Suffice it to say, it has a significant impact on the cell biology of the yeast and of its cellular adaptation to these high concentrations of ethanol. We'll speak about that a little bit later. During this process of actually consuming the sugar, it also consumes nitrogen. So you get into an area where nutrient depletion is becoming a real impact for these yeasts. Not only are they not growing and there's a high concentration of sugar around, there's still a low pH, there's no oxygen, the temperature is often low, and it's running out of nutrients. So if we look at all of these things, these are basically the fermentation stresses that the yeast has to cope with. So we're asking it to adapt to low temperatures, to high acidity, to osmotic stress, to high concentrations of sugars, no oxygen, no nutrients, and then the ethanol levels that increase. We wanted to make a great wine. The yeast doesn't care about what we want. What it wants to do is to protect its own genetic material, to grow, and to survive in this environment. So what it does is it adapts to all these stresses, and goes through physiological and metabolic changes so that it can actually perform those functions. So it senses these specific stresses serve as stimuli for the yeast, which it can sense. Those proteins that we had on the outer surface, those nice little dots that I had on the outer surface of the yeast, some of them are sensing proteins in which they can actually sense the environment that the yeast finds itself in. It internalizes that signal, and that signal then becomes internalized into the yeast itself, and it can go to adapt that central mechanism that I spoke of that underscores the function of the yeast. So it can change its transcriptional profiles, it can change or adapt its proteins by looking at how much protein is actually present and where they go within the cell. We'll talk about that a little bit later. And then it adapts metabolically as well. Consequently, there's an entire cellular adaptation for the yeast to cope with this. And hopefully in this whole process it still makes a nice wine for us. What we want to do is to see what this adaptation is all about, so that we can select for or come up with new strategies in which we can treat fermentation so that this process is user-friendly for the consumer at the end of the day, so that we don't get into the problems that are associated with fermentation stresses. So what are the impacts of these fermentation stresses? So, well, we mentioned one already, and typically you have this lag phase. We have to actually wait until the fermentation gets going. Now, in most instances, it will start within a day or two. But in some instances, winemakers get a little bit antsy if after 2.5 days or 3 days the fermentation isn't really taking off. Then they start fiddling with temperatures and all kinds of different things to try to get the fermentation to go. So there are significant problems with a slow start of a fermentation. So this increased lag phase can potentially translate into inefficient fermentations that can lead to stuck or sluggish fermentations. We see these increased lag phases in wine, ice wine, and sparkling wine productions. I'm not going to go into ice wine and sparkling wine production, but they are much more severe than for regular table wine production. These inefficient fermentations or stuck or sluggish fermentation is often caused by a delay in the sugar utilization, nutrient uptake during the fermentation process, and often they can take weeks to actually resolve themselves unless the winemakers are proactive and try to kickstart it in a different way. Ultimately, this affects the quality of the product at the end of the day. You get off-flavor production often associated with sulfur compounds that are being produced that would lead to significant drop in quality of the wine. And there can also be the development of spoilage compounds and all the off flavors or undesirable flavors associated with these. So these all negatively impact the quality of the product at the end of the way. And consequently, there's an inefficiency in the winery. The cellar operations suffer, and what is affected at the end of the day is that the winemaker has to report to the boss man and say that we're losing money. So consequently, what we want to do is to see whether or not we can understand these issues a little bit more at a fundamental molecular basis, and if we can come up with new strategies to overcome this problem, and that winemakers can sleep better, and consequently the owners as well. So how do we go about actually measuring the yeast's response to these fermentation stresses? So there are a few things obviously that stand out. As we can, at a molecular level, let's start with a metabolite level first. The products or the nutrients or precursors of these reactions can be measured. So you can, for example, look at the amount of sugar that's still present in a fermentation, or the precursors for some volatiles, or for some flavor compounds. You can look at those precursors and see whether or not the yeast is actually consuming them. And so during a fermentation, you would expect that sugars would get less. And so we can go and measure the sugar during a fermentation to see whether or not that is the case. On the flip side, we can say, well, let's see how well this is actually running, so let's go and measure a product of these reactions. One of those could be, for example, ethanol, see how effectively ethanol is being produced. That would be a readout to how effectively the fermentation is running. So we can do those metabolite screening in which we just look at the juice of the grape must as it's being fermented. What we can also do is we can physically just go and count the yeast and see how well they actually grow, and then consequently get a measure of if they did grow, if they get to levels where we expect them to be. So this is something that we can very easily do. What we want to understand, though, is how does the yeast adapt at the molecular level? So we have to dig in a little bit deeper. We have to use more of a molecular analysis approach to this. So what we can go and do is, and we'll speak of a little bit later, is go look at these RNA levels. In other words, how the DNA is transcribed into RNA, how much of that is taking place, and does it take place effectively. What we can also do is we can go and measure protein presence or absence. Is it there or is it not there? And if it's a protein that's supposed to, for example, function on the plasma membrane, is it actually on the plasma membrane when we think it's supposed to be there? Or is it somewhere else? So those are all measures or analytical tools that we can use to go and study the molecular response of the yeast to fermentation conditions. So what I'm going to talk about now is basically one of the PhD students in my lab, currently a postdoc, started to look at some of these nutrient transporters, specifically hexose transporters. In other words, the main transporters that's needed to carry sugar into the yeast during fermentation. So how do we actually see this protein? Because he wanted to know whether or not these hexose transporters are on the plasma membrane when it's supposed to be there. So how can we go and visualize this? This is a microscopic organism, and it's very small. So we need something that we can actually detect with a microscope. So what we can do is there are these funky proteins around in nature that can actually fluoresce when you shine a UV light on them. So consequently, with a fluorescence microscope, you can actually shine UV light on a specimen, and then everything that has a fluorescent protein will shine. And consequently, we can see where that is within a cell. So what we can do, and what we did do, is that if you can imagine that this would be the chromosome of the yeast, the DNA of the yeast, and this is a gene that encodes a hexose transporter, HXT3, which, by the way, is the most important hexose transporter for winemakers because it takes up most of the sugar throughout the entire fermentation process. Winemakers love this guy. So what we do is we actually generate a DNA fragment that can very specifically insert at the back end of this gene and consequently fuse your hexose transporter gene with your green fluorescent protein, and consequently you get a fusion protein, or a fusion gene that looks like this. If that is transcribed and expressed, you see the hexose transporter is actually inserted into the plasma membrane itself, and you've got your green fluorescent protein that's hanging inside the cytosol. Right? Now we can actually go and look at this and see this even under a microscope. It still performs its function very easily by taking up glucose and fructose into the cell. So there's nothing wrong with the protein. It just now has something that we can actually go and visualize. So this young man is Chris Snowden. He finished his PhD in my lab earlier this year. Early last year. And he's been a postdoc there for a couple of months. And he was interested in understanding how a group of proteins that we study in the lab called the VIT30 complex, how they actually function in regulating the adaptation of the yeast to a changing in nutrient conditions. So it is known that a group of genes, the VID or GID genes, encode a group of proteins that actually form a complex within the cell. Okay? And it is known that this complex per se is involved with the adaptation process of the yeast to changing nutrient conditions. We call this the VID30C because one of the components of one of the proteins of this complex is actually called VID30. So Chris wondered whether or not this protein complex is actually involved in the regulation of hexose transporters, and there were 2 hexose transporters that he was interested in. I won't explain in detail why that is the case. That's going to take us a while. So what he did is he fused H67, which is a high affinity hexose transporter. This protein functions at the end of a fermentation. In other words, when sugar concentrations get really low, where you want to take up that last little bit of sugar that's around to basically get the wine to be dry, you want H67, which is a high affinity hexose transporter, to be around and be active. So Chris wanted to know whether or not our complex, the VID30C, is actually involved in the regulation of H67, this very important high affinity hexose transporter. And he studied this in the context of nitrogen starvation. So he went and he added a green fluorescent protein at the back end of H67, and then grew the cells in a low concentration of raffinose, actually in the absence of glucose, which would induce the expression of H67, and consequently you can see this very bright fluorescent ring that surrounds the cell. That's the plasma membrane with this transporter in it. Then he starved the cell of nitrogen, which is often the case at the end of a fermentation. And the interesting observation that he had is that he saw that some of this fluorescence with time internalizes into the cell, leaving the plasma membrane, and consequently becomes lighter, which means it's being degraded. But after nitrogen starvation, he didn't see any of the hexose transporter on the plasma membrane anymore, which is an indication that if you run out of nitrogen at the end of the fermentation, your high affinity hexose transporter is also going to go missing. So consequently, the fermentation is not going to get dry as effectively as you would want it to be. So what he then did is he went and he mutated the VID30 complex to see whether or not our protein complex of interest is involved in regulating this. So he knocked out some genes in the VID30 complex and did exactly the same experiment. And what he saw after 24 hours, or up to 24 hours of starvation, that this hexose transporter is actually still persistent within the plasma membrane, indicating that this complex that we're studying is actually involved in regulating the nitrogen-dependent turnover of this high affinity hexose transporter that is crucial at the end of the fermentation. So this is the kind of fundamental knowledge that we now would want to actually use and apply to the wine industry, to see whether or not we can build this selection process to get yeast that can actually have these hexose transporters on the plasma membrane at the end of the fermentation when it is really needed. We are now starting to understand how that works at a fundamental level. So now we can go and actually devise strategies how to come up with an idea of making a yeast strain that can be more effective towards the end of a fermentation. So this is for H67. This is a high affinity hexose transporter. So Chris, being the great scientist that he is, decided, well, I'm going to do the same thing with the big boy. In other words, with the H63, the one that functions at the front end of the fermentation. And he said, well, if we actually study this, in which we shift the yeast cells from glucose to ethanol, glucose being the high concentration of glucose that's present at the beginning of a fermentation, and then ethanol when we actually run out of sugar, when sugar is not needed anymore, do we see that this hexose transporter is turned over again in a manner that's dependent on the VID30 complex? And yes, he found that when he grew the cells in a high concentration of ethanol, this is a little bit lighter than H67, you can see the H63 GFP in the plasma membrane, and then as he actually shifts the cells into ethanol and leaves it in there, the hexose transporter leaves the plasma membrane, gets internalized, and it's degraded, and as soon as you knock out the VID30 complex, that actually is a little bit more stable. So it means that this complex of proteins is involved in regulating hexose transporters and their turnover regardless of the conditions. It's not just in one condition. It's in a variety of different conditions. So what we're learning from this is that we're starting to fundamentally understand when these hexose transporters are turned over and what are the proteins that's involved in that. And we can consequently use that knowledge, or that's what we're trying to do now, to build or select for yeast strains that have these hexose transporters stable in the plasma membrane for longer. so they can more effectively ferment. And now I'm going to shift gears into that fortuitous project that I spoke about, that we kind of backed into. When we looked at the Vit30 complex that we spoke of earlier, one of the components of this complex is called Mo1, which we have no idea what the function of that is at this stage. An interesting thing is that it was also found to be present in another protein complex, in which there were 3 more proteins. One of them being ubiquitin, and we'll speak about that at the end of the talk, and then also this protein called ETP1. And because this protein was present in 2 complexes, we had this harebrained idea, Chris and I, that we might want to go and have a look at this additional complex. And the thing that triggered our interest was the fact that ETP1 is actually, its transcription was induced by ethanol. So we thought that, okay, if its transcription is induced by ethanol, then maybe it's activated in the presence of high concentrations of ethanol. There's ethanol being produced in the fermentation. Maybe it has a function in that environment. So we thought, okay, well, let's go and have a look at this. So if we look at ethanol tolerance, just quickly, 7.5% ethanol is generally regarded in the literature as being ethanol stress. At this concentration of ethanol, it really impacts the fluidity of the membrane. Right? So this is one of the problems that the yeast run into during fermentation, specifically later in the fermentation, is that the structure of the membrane is affected by ethanol, and consequently the efficiency by which nutrients are taken up into the cell is affected by the high concentrations of ethanol. That's what the yeast has to combat. It also denatures proteins, both proteins that are in the membrane and soluble proteins that's inside the cell. So this is not a friendly environment for the yeast to be in. It tries actually to adjust to that. So this process, this ethanol toxicity greatly impacts the viability of the yeast cell. What the yeast does in response to that is that it has the ability to change the nature of its membrane. It can't make new membrane, but it can actually change the nature of the membrane. So it tries to do that to adjust the membrane fluidity so that it doesn't lose all the function of the membrane itself. And then it also increases the transcription of what's known as chaperone proteins. These are proteins that help other proteins fold correctly. So combating this denaturing step of ethanol, it basically transcribes proteins, these chaperone proteins, and in yeast they're known as heat shock proteins. So it increases the transcription of these genes. So we thought, okay, with this in mind, let's go look at ETP1. And we, a little while ago, we published a paper on ETP1 and how it is actually needed to adjust to ethanol in the environment. So what we did here is we grew the parent strain and a couple of mutants that are part of the ETP1 complex and the ETP1 mutant itself. And we did basically a growth assay in which we had a rich medium that we supplemented with different concentrations of ethanol. And in all instances, you'll see that the wild type and these mutants, the first 2 mutants, grow fairly similarly. In the absence of ethanol, there's no difference between all 4 of the strains we tested. But as soon as we add ethanol, the ETP1 mutant started to struggle. It doesn't grow as effectively as the wild type strain does. And the same thing goes for when we actually change the carbon sources around. If we change the glucose in the media to glycerol and ethanol, or just glycerol, as soon as you only have ethanol or ethanol combined with something else, the ETP1 mutant struggle. So this told us that, okay, it's needed for the adaptation to ethanol in the environment, right? So we said, well, we know these heat shock proteins are transcribed at a high level when there's ethanol stress around. Let's see whether or not our ETP1 protein is actually needed for that transcription. So we did a simple assay in which we grew the parent strain and the ETP1 mutant in the absence of ethanol stress, and then we extracted the RNA, and we wanted to see whether or not these genes are actually transcribed in the wild type and the ETP1 mutant. And we see very little. Here you can't see any transcription of these 2 heat shock proteins. If you overexpose the blood, you see a little bit. However, when you go and you actually do ethanol stress, you apply the 7.5% ethanol to the growth conditions, you see that there's a high level of transcription. It's not so high on this image. If we look at the overexposure over here, you see a high level of transcription of HSP12 and HSP26 in the wild type, but there's a lot lower level of transcription in the ETP1 mutant, as indicated over here. If you do densitometry on these, in other words, if you measure how intense these signals are, you can see that in ethanol stress, there's about a 35% level of expression of both of these genes in the ETP1 mutant compared to the wild types. So we know that this specific protein is needed for ethanol tolerance and that it affects the transcription of these heat shock protein genes. Chris, being the hexose transporter guy, said, well, let's go see whether or not H63 is actually, its turnover is affected by ETP1. And surely it was, just like you saw with the VIT30 complex. You could see turnover in the parent strain, but in the ETP1 mutant there's a delay in the turnover process. So we know that that's involved in that. So all of this work was done in lab yeast. Right? So this is our typical workhorse that we use in the laboratory. But we know at the end of the day that lab yeast and industrial yeast are two vastly different beasts. You get an idea in the lab yeast of what's happening in the industrial yeast, but you still have to go and confirm it. Just because industrial yeast has been adapted so well over so many generations, thousands of years to fermentation conditions, it does respond differently than the lab yeast does. So we thought, okay, this is going to be an easy thing. We're going to make a knockout of ETP1 in the M2 yeast. We're going to make a homozygous mutant. In other words, this is a diploid organism. It's got 2 copies of every gene. We're going to knock both ETP1 genes out. And we're going to make a heterozygous mutant in which we're going to knock out 1. Then we're going to do Chardonnay fermentations. And what we're going to see is that there is actually ethanol tolerance. It's needed for ethanol tolerance. So what that would mean is that we expect the homozygous mutant to ferment really well up to about a third into the fermentation when ethanol starts to build up, and then it's going to start croaking. Right? We won't be able to deal with the ethanol anymore. It's going to stop fermenting at that stage, and we expect to see that there's going to be a high level of transcription of HSP genes. And we were wrong. It's totally not what happened at all. Showing that lab yeast and industrial yeast are vastly different beasts. And that's the nature of the work of a scientist, is you think you know what you're doing, and then you realize, I've got to go and test everything again. So, enter Ashley Hillier. She's a great lab person, did phenomenal work with this project. So Ashley actually went and she made the heterozygous mutant and the homozygous mutant of ETP1, and proceeded to do Chardonnay fermentations. We thought, okay, Terence Van Rooyen of Niagara College is going to give us the Chardonnay juice, and we're going to do some fermentations. This is what Ashley did, and this is the daily weight loss that she actually measured in the fermentation. The blue would be the wild type, in which you can see a typical delay, a lag phase that would happen, and then the spike over a couple of days in the beginning of the fermentation of weight loss, and then it peters out towards the end of the fermentation. The heterozygous mutant in purple over here, where it's still got 1 intact copy of the ETP1 gene, pretty much looks the same as the wild type does. So it means that this one gene can actually still perform its function. As soon as we knocked out both copies of ETP1, at day 2 it basically stopped fermenting. So right in the beginning of the fermentation, where there's hardly any ethanol around. It already had a phenotype, it can't ferment. So our whole thinking about this is ethanol tolerance related went out the window immediately. So then we had to figure out, okay, why does this happen here? We've got no idea really what this problem is. All the data we generated thus far indicated that this was actually not going to happen. So Ashley went ahead and she did a proper growth curve, a proper fermentation profile, in which you can see that the error bars on these fermentations are very little with the ETP1 meat. We get that all the time. And then with the wild type, this is what she got. This is Chardonnay fermentation and then the weight loss over an 18-day period. So we see even at day 1, we see that there's still a similar amount of weight loss in the wild type and in the ETP1 mutant, but then pretty much after that we see that it starts to divert. And while the ETP1 mutant continues to ferment, it does it very slowly in comparison to the wild type. So she went and she actually did some metabolite analysis on day 18, collected samples of the juice and did some metabolite analysis. and compared that to the metabolites that you would have within the Chardonnay juice itself. So this would be fructose and glucose in the Chardonnay juice, and you can see that we actually stopped the fermentation a little bit too early because there was still a little bit of fructose left in the wild type and almost no glucose. But what is obvious here is that there is still fructose and glucose left within the ETP1 mutant. So consequently, it can't ferment as effectively as pretty much what the weight loss predicted it to be. Even though there is a statistical significance in the difference between the levels of glycerol between the wild type and the ETP1 mutant, these levels are really small. It's almost insignificant in the sense of difference. However, as we can predict from the ethanol, or from the glucose, we can infer that there would also be a difference in ethanol, and that was indeed the case, that the wild type would produce more ethanol than the ETP1 mutant does. So this pretty much matches up with what we saw in the fermentation trial. We also did, with the help of Barry Shelp at Guelph, we did some metabolite analysis of specifically amino acid analysis using HPLC of the juice, of the Chardonnay juice, and then also of the N2 strain and the ETP1 mutant at the end of the fermentation. And the take-home message here is that you always see that the wild type, the M2 strain, would use quite a bit of these amino acids, so those levels would always lower than the, in most instances, lower than the what was present in the Chardonnay juice, and that the ETP1 mutant, these light green bars, are in most instances higher than that of the wild type, indicating that the ETP1 mutant had a problem in actually taking up and metabolizing these amino acids. Right, so not only does it have a problem with sugar metabolism, it also had a problem with amino acid metabolism. Right, so it couldn't take up these amino acids as effectively. So we were baffled. What are we going to do now? How are we going to figure out what the function of this ETP1 is, and how does it actually impact the cell itself? So we knew that it was involved in a transcriptional context. We knew that the ECHOC proteins were transcribed to a lesser extent in the wild type, or in the mutant versus the wild type when we did our assay. So we thought, okay, we know that there's a problem right at the beginning of the fermentation. Why don't we do a transcriptional analysis right around this starting point, and then also one later on in the fermentation, and we see how the genes in general are transcribed in these two strains, in the M2 strain versus the ETP1 during a Chardonnay fermentation. So this is the real thing as you would have within the industry. So we actually went and did 3 biological triplicate of the ETP1 mutant and of the wild type. So these are 3 independent fermentations, not the same fermentation sampled 3 times. And then collected samples from them at these indicated time points, extracted the RNA, and then sent it off to Toronto to be analyzed using microarray analysis, so that we can see how all the genes within the yeast cell are transcribed at a given time point. Right? So this is a global view. This is not just one gene at a time. So we see a phenomenal impact of this protein on the transcriptional level for the yeast. The p-value for the statisticians around the p-value that we used as a cutoff was 0.05. So all our genes that we analyzed had to fall within this statistical parameter, so that we know we're looking at the real thing and not at artifacts. And then we also looked at all genes that were actually changing more than 2-fold. If we didn't make this cutoff, we didn't analyze those genes. So we were very selective and strict in that. So if we compare the number of genes that were actually expressed in the ETP1 mutant compared to the wild type, we found that in all time points there were significant responses. 10 hours into the fermentation, we can see that it's already almost 250 genes that changes expression, which is significant, keeping in mind there are 6,000 genes roughly in the yeast genome. 24 hours in, we see approximately 600 genes change, 860 2 days in, and that almost doubles to 6 days in. So this is mind-blowing, because what do you do with all this data? Right? How do you infer function from something like this? You've got so much information coming in, you've got a massive amount of genes that you now have to look at, and this was indeed a daunting task for Ashley. So what we typically do with this is we start breaking it down into categories or functional categories of groups of genes that are related to each other. And I'm just showing a couple because I'm going to be speaking to two of these in a little bit, but there are many more categories that you can actually group these genes into and consequently see whether or not there are families or groups of genes that respond specifically to your mutation. That would give you an idea of what the function would be. So what we did see across all time points, specifically from 24 hours onwards, that genes that are associated with amino acid and nitrogen metabolism are significantly affected. Right. So this would also explain why we saw the amino acid data, the inefficiency to actually metabolize amino acids. This would be partially explained by these changes in gene expression. Remember, this is things that these genes are now responding differently to the wild type, to what is supposed to be happening. This is what's happening in the mutant. Something that I want to talk about in a little bit more detail is cold and anaerobiosis. So there are groups of genes that very specifically respond to this. And remember, we spoke about the stresses that the yeast actually experiences at the beginning of the fermentation, where cold and anaerobic stresses actually are two of them. And there are specific genes that actually respond to this. The interesting thing is that they're always or down-regulated, specifically in the ETP1 mutant. There's more down in the down-regulated column than there would be in the up-regulated column. And this is a specific trend that seems to last. The other thing is actually genes that are associated with the cell wall, and specifically one component of the cell wall, where we see that there's a specific upregulation of genes that are involved in cell wall metabolism, right? And these are fairly high numbers. So we're going to talk about these in a little bit more detail. So let's talk about the yeast cell wall just for a little bit. We used to thinking about the yeast as a budding yeast. This two-dimensional images or three-dimensional image is what jumps into mind when we think of them. But we also have to pay attention to what is actually the makeup of this specific cell wall. So this would be the inside of the cell. This is a higher resolution transmission electron micrograph, where this, you might not be able to see this, but there's a double membrane or plasma membrane that's here, and then there's a gap between the periplasmic space that's between the plasma membrane and the cell wall, which is this outer component of the yeast itself. So we have to actually look at these components in a little bit more detail. So we have the plasma membrane, and then associated with the plasma membrane, you would have a variety of different polymers that we find extracellular to the yeast. These things are made within the yeast and transported to the outside, and then they are linked to the plasma membrane in a variety of different ways. So the polymers that we have are chitin, that are very specific, specific to yeast, and then also beta-glucans. These are all sugar polymers that are actually on the extracellular surface of the yeast, and they are crucial for the yeast survival because they actually provide structural support for the yeast. So for it to tolerate hyperosmotic stress in ice wine fermentation, for example, or in wine fermentation, it provides the structural support for the yeast to survive at the end of the day. So this is critical. These polymers are sugar polymers, and they link to each other either biochemically or covalently, or via other proteins called manoproteins that can either link these proteins specifically to each other, or the manoproteins can also be anchored by these black lines to the plasma membrane itself. So if you think about the yeast is constantly growing, it means that the cell wall has to constantly be remodeled and to grow as well, to be reshaped as well. If you take this yeast and you actually inoculate an hyperosmotic environment such as ice wine juice or grape juice, it's an hyperosmotic stress, all the yeast cells shrink. They become smaller to be able to combat the hyperosmotic stress better. To do so, they have to remodel the cell wall as well. So there's constant changes that actually takes place to the cell wall so that the yeast can actually adapt to different environments. And so the cell wall is critical for the yeast viability. So there are a variety of things that impact cell wall that the yeast would experience right at the beginning of the fermentation. Temperature and hyperosmotic stress and anaerobic environments would be the two that are of greatest importance. So what do these things do? Or if we think about those stresses, cold and anaerobiosis during the wine production process. So we know that the temperature of the grape juice in the fall can be low. We know cold soak takes place in the wine production process quite often. And we also know that whites are typically fermented in colder fermentations. We know that oxygen is also a major threat to the wine industry, and consequently winemakers go through a great extent to actually protect wines against that, to prevent oxidation of flavors, of flavor compounds. There are typically low levels of this dissolved oxygen that are present during the fermentation process. And to top it all, whatever levels there are present are very quickly scavenged by the yeast. It scavenges that oxygen very quickly. Some winemakers would actually oxygenate, make use of oxygenation during the wine production process, whether consciously or subconsciously. Pump overs can be ways in which you actually oxygenate the fermentation, but some people actually go as far as doing micro-oxygenation to try and vitalize the fermentation process. Suffice it to say, the yeast very quickly consumes this oxygen during the fermentation process. So, how does this impact, just to restate, how does this impact the yeast, this cold and anaerobic conditions? So, just like we had with ethanol tolerance, there's a decrease, or like we had in ethanol tolerance, there's a decrease in the fluidity of the plasma membrane, and consequently there's a decrease in membrane function due to anaerobic conditions or cold specifically. In the absence of oxygen, the yeast can't make new membrane, but what it can do is it can alter the existing membrane, and it does so. It changes basically the characteristics of the lipids that are in the membrane, so that it would actually become more fluid and consequently be more effective. Also importantly, it also remodels the cell wall, like we spoke of a couple of slides ago. It alters not only the components that are in the cell walls, in other words, those polymers we spoke of, but also the proteins. It can change that very specifically in relation to to cold or anaerobic conditions. So there are a group of manoproteins that are very specifically transcriptionally regulated, induced actually in response to cold and anaerobic conditions. And these are called the DAN or TIR or POW genes. And they're all related, a big happy family. They all are induced when the yeast actually gets into anaerobic or in cold conditions. And we're going to speak of them in a little while. I don't know if I want to go through this. Okay, so the yeast senses the environment and consequently changes. So if you can imagine that this would be a yeast cell, this is a nucleus where transcription takes place, and we have some kind of fermentation stress, say cold or heat or some kind of temperature shift that serves as a stress. There are sensors in the membrane of the yeast that sense that. These are proteins, these bubbles that are colored, all proteins. Once these sensors actually sense these differences in the environment, it internalizes that and activates a signaling cascade. In other words, a whole group of proteins that responds to that. And we're not going to go into detail with that. Suffice it to say, there's one protein in this instance, MPK1, that's really important for that protein, for this response to take place. And we can go and measure that protein very specifically. Once this protein is active, there are a group of genes that respond that are actively transcribed. So when we look at our data that we generated, our microarray data, and we looked at all these different time points, 10 hours, 24 hours, 48 hours, and 144 hours, we found that all the genes, or some of the genes that are actually encode the proteins of this cascade, are misregulated in the ETP1 mutant. There's actually more of them. Not only is there more of them, this is the terminal kinase. This is the one that we said MPK1 that's important. Once this is actually active, the entire pathway is active. So we see there's more of these signaling proteins are activated. So our first response is, wow, okay. So now do we see the genes that are actually downstream of that? In other words, all the genes that are needed for remodeling of the cell wall that we spoke of earlier, are they also regulated? And we see that there's a whole, this isn't all of them, this is just an example. But there's a whole slew of genes that are associated with the production of compounds that are within the cell wall that are all upregulated in a manner that's dependent on the absence of ETP1. Remember, this is the mutant that we're looking at. So now that the ETP1 mutant is not there, we see an increased activity or increased level of protein or level of gene expression of these regulatory genes, and then all the genes that are subsequently also under its response. Right? So this is under its control. So this is actually a fairly significant finding. We have not seen that before, implying that there is a regulation of the cell wall pathway in the beginning of fermentation, which makes a lot of biological sense. What we can do as well is we can actually go and detect the protein levels of MPK1, for example. So there are these things called antibodies that very specifically bind to that protein. So what I'm going to show you here is basically the wild type, the parent strain, and the ETP1 industrial yeast, in which we've extracted protein from very specific samples of the microarray data set. So this is 10 hour, 48, 144 hour sample points. We extract protein as well as RNA. The RNA was used for microarrays. And then we analyze the proteins here. What we also did as a control is we basically stressed the cell wall with a compound that is known to stress the cell wall. And we analyzed in a rich medium the absence and the presence of this stressor. And in the ETP1 mutant, we can see there's a little bit of the MPK, in the wild type strain, there's a little bit of this MPK present in the absence of the stressor, and that actually increases in the presence of the stressor. So we know that it responds the way we think it's responding. When we do the same thing for the ETP1 mutant, we see the same response, but to a lesser extent. So there's less of the protein, which means that even though the gene is transcribed, there is less of the protein, indicating that ETP1 is needed for the stability of this protein. If we actually go and look at the fermentation samples, you see at 10 hours, we could actually detect MPK1 in the fermentation samples, and we couldn't do so at 48 and 144 hours, but at 10 hours we could not detect MPK1 in the wild type. In the ETP1 mutant, indicating that our protein of interest, this MPK1, is actually its stability is dependent on ETP1. Once ETP1 is absent here, we can't detect the protein. So we've got a very significant regulator that is controlled by ETP1, which we did not know before. We then moved on and we looked at high concentrations of sugars. I promise you this is the last slide of this nature that I'll show you. So this is actually of interest to most of the Canadian wine industry because it deals with high concentrations of sugars and also anaerobic stress. High concentrations of sugars specifically because of the ice wine industry. So there is this signaling pathway that allows the yeast to respond to this. There are sensors in the environment that would sense the presence of high concentrations of sugar, and also recently in anaerobic conditions. This signal would be internalized and relayed to a kinase, a very critical kinase within the yeast called HOG1. Right. This kinase is then pretty much like MPK1 in the previous section, is then responsible for a transcriptional response within the nucleus itself. Without HOG1, we would not be able to make ice wine in Ontario. Because this allows the yeast to actually survive in the presence of high concentrations of sugar, because it actually goes into the production of glycerol and consequently can survive. When we look at HOG1, what it also does, it responds to anaerobic conditions and it transcribes all these genes known as POW genes that we spoke of earlier, those manoproteins that are part of the cell wall. So Henny van Vieren in his lab actually looked at one of these POW proteins, and he could find that when he tried to detect them in a variety of different conditions, he found that this is actually present in low oxygen, concentrations of oxygen. You could find the protein, and also in cold, you can actually find this protein. He tried to detect it during the fermentation process as well, and you could see that there are a variety of, from 24 hours onwards, you could see this in the fermentation. You can actually detect the protein. So we know it is actually transcribed during fermentation. The same thing went with when he tried to study other POW genes. You could actually see the proteins are present at different time points of sugar fermented during a Chardonnay fermentation. So we know that these genes are actually actively transcribed. What was recently also showed is that HOG1 is needed for the transcription of these POW genes in the absence of oxygen in anaerobic conditions, which is what you would find in the fermentation. And what Fred Winston in his lab also showed is that there's a transcription factor, UPC2, that's needed for this activation. If he knocked out this gene, you didn't see the transcriptional activation. So when we actually look at our data set and we look back at this, we could see that almost all the POW genes are down-regulated significantly in our data set. So consequently, there's a massive transcriptional response that we see here. We also see that that transcription factor that we spoke of earlier is also down-regulated significantly. So consequently, all these proteins that are needed for remodeling of the cell wall in anaerobic conditions, we found to be needed in the fermentation as well, and that its transcription is actually needed, regulated indirectly, more than likely, by ETP1. So what we basically say here is that there's ETP1 is involved in the regulation of all these POW genes, or this family of POW genes, and also of that transcription factor we spoke of earlier. Fred Winston did show in his lab that HOG1 was actually the kinase that's responsible for regulating all of this in anaerobic conditions. So we thought, okay, well, let's go and see whether or not we can detect HOG1 in our time points of our samples that we collected for the microarray analysis. And what we did here is we basically extracted the protein and see if we can actually detect HOG1. What is very clear is that as a control, again, we used sorbitol as a stressor, which would be which would simulate hyperosmotic conditions, and we see that the HOG1 protein is present both in the M2 parent strain and in the ETP1 mutant. It's present at high levels. However, and this is the interesting bit, in the fermentation process itself, we see HOG1 is actually present in the wild type at fairly high levels right throughout the fermentation process. However, it's absent in our ETP1 mutant. Consequently, this would explain the absence of HOG1 and the downregulation of that transcription factor would explain why these levels of transcription isn't higher. So we know that two of the most central regulators in the yeast cell when it comes to adaptation to a variety of different conditions, HOG1 and MPK1, is actually not present in the start of the fermentation when ETP1 is not there. So this gives us new fundamental insight into how the yeast actually adapts to different environmental stresses during the fermentation. So the conclusions that we can make from this is that ETP1 is actually needed for a normal fermentation to occur, and it significantly impacts the transcriptional adaptation process. We've identified some regulatory proteins that are affected in this, that are regulated by ETP1 specifically. HOG1 and MPK1 would actually be those two regulatory proteins. We have some evidence in our lab that there are actually other proteins as well, but it is way too preliminary to even speak about those. We see that there is some readouts downstream of these regulatory proteins not being regulated effectively, like the POW genes and the misregulation of the cell wall genes. And lastly, we've got some insight into what ETP1 does. If you go look at the ETP1 protein specifically, it has the characteristics of a protein that binds ubiquitin, and that is needed for protein turnover. So we're not going to go into discussing that mechanism, but we suspect that ETP1 is needed for the turnover-related process of other target proteins. We don't know what those proteins are, and that would be the subject of our investigation as we move forward. So what we actually know from this is we've now identified new players early in the fermentation that we never knew of before, how the yeast actually adapts to fermentation-related stresses, and now we can use that knowledge to actually develop new strategies for fermentation, more effective fermentation adaptation right in the beginning of the fermentation process. The people that actually did the work that I have to thank here, Chris Snowden, Stephanie Hughes, and Peter Polichuk for the original work on the ETP1 in the lab yeast. Chris did all the work on the VIT30 complex as well. For the work that we did with ETP1 in the industrial yeast, Ashley Hillier did most of that work, and then we had a bioinformatician in my lab, Kirill Besanoff, that did the statistical analysis of the microarray data. He was a kingpin in that. The people of my collaborators that actually played a role in this project specifically is Barry Shelp, did the amino acid analysis for us, and we did some of the sugar and ethanol analyses in the lab of Hung Lee. And then Terence van Rooyen gave us the Chardonnay juice, very gratefully. And the people that actually paid the bills, University of Guelph, OMAFRA, and the Genome Canada were the ones that actually footed the bill for these guys. Thank you.
Do we have time or some questions from the audience?
I would just ask, I struggle to keep up, and what would be the next steps for you in terms of practicality and the impact to winemakers?
Okay, so that's always the problem that I have, because we first start with the fundamental stuff and then we apply that. So we obviously know that the absence of ETP1 now relates to a significant problem. So now the flip side of that would be is, okay, if we can stabilize this protein in a yeast, in other words, that ETP1 is present, now do we see the opposite effect? Do they actually combat stresses more effectively? Right, so now we basically turn that question upside down. And that's often the case. And so there are a variety of ways in which you can do that. We can do that, try to do that through selection, which is a really hard process. What we can also do is we can go and actually overexpress that gene using strategies that's user-friendly and that's not genetic manipulation, and to see whether or not we actually can see the opposite effect happen. And then test that in fermentation trials. This guy first.
Have you done much work on the effects of different solids in wine, like leaving more solids after racking and on the effects of stresses on yeast?
We have not done that. No, we have not done that. No, that would be a nice project though. Yes.
In normal yeast cells, is there a general decline of ETP1 as fermentation progresses?
We have not been able to detect the protein very effectively, which is something that is kind of expected. We think that it is an E3 ubiquitin ligase, so it's a protein that adds those ubiquitin proteins I spoke of onto other ones. They're notoriously unstable in the sense that they've got typically have short half-lives, and consequently they get turned over themselves as well. If we stabilize that protein, we don't know what the readout is going to be. So to answer your question, we have not been able, if we tag ETP1 with GFP like we did with the membrane proteins, we can't detect it to start off with. We know that it's in the genome, we can detect that, but we can't detect it in the yeast itself. Now that's often the case because of low expression levels. It's just below the level of detection. That we can only detect up to a certain level and not lower than that. So what we would like to do now moving forward is to overexpress ETP1 to see what happens, stabilize the protein and see what happens. That kind of stuff would be where we would be going with this. Good question, though.
George, in the earlier work on the hexose transporters,
yes,
throughout the fermentation and then when they're put under nutrient deficiency stress and the transporters get internalized and degraded, where are you in terms, and of course my interest in this and winemaker's interest in this is, near the end of the fermentation,
yes,
as you pointed out, that's when nutrients can be depleted, and that's where often, stuck or sluggish fermentations occur, and if you don't have those transporters in the membrane, you can't get the sugar into the cell in order for the yeast to ferment to dryness. So where are you at in terms of understanding or reversing that regulation, in terms of, again, knowing targets to look for the next generation of yeast that get developed for the industry?
Yeah, we're quite a ways from being at that specific level. We have got a rough idea of where these turnovers start, but the molecular mechanism that underscores that's a significant hurdle that we have, is that there's so much difference between the industrial yeast and the lab yeast that we basically have to reinvent the wheel in the industrial yeast. Even though we have the sequence of the lab yeast out, and then now the industrial yeast has also been sequenced, there's over 60,000 different point mutations compared between the two genomes, and the industrial yeast has an extended genome as well. It's got more genes. So the mechanism of regulation is not the same. So
Especially during those.
especially during the high stress conditions right at the end of the fermentation, right? So we've identified some, at least when these things start happening, but into developing a new yeast to actually overcome that, we don't understand the mechanism enough here to go there. So, as you know, it's going to take us a couple of years to actually get to that.
It just means we still have lots of work ahead of us.
Yes, I still have a career.
Are there any other questions? Were there any online? No. Okay. Well, please join me again in thanking George for a stimulating talk today. Lots of interesting molecular biology, but hopefully we all also could appreciate the practical applications that come from this type of work in the future. So thank you, George. And here's a small token of
Oh, thank you. Thanks very much. Wow, that's surprise. Thank you.